Hi, my name is Naila Rabbani. In this video, I would like to tell you the biomarkers and its interpretation. We chose to study glycated, oxidized, and nitrated protein damage biomarkers because these over time accumulate and give you the full picture of what is happening to the metabolism in different conditions. So I would like to tell you what these biomarkers tells us about autism. So our blood test for autism is based on plasma protein glycation and oxidation biomarkers. In our discovery phase study in 2018, the minimum features of our diagnostic algorithms was 4 biomarkers, although we measured 15 different adults. The 4 features were plasma protein content of N-carboxymethyllysine, CML, N-carboxymethylarginine, CMA, and 3-deoxyglucosone-derived hydroimidazolone, 3-DGH, and 4th dityrosine, DT. The first 3, CML, CMA, and 3-DGH, are protein glycation adducts, advanced glycation end products, AGEs, and the last one, dityrosine, is a protein oxidation adduct. These adducts occur on multiple sites of proteins, but mostly albumin in plasma. So we measure the total adducts of each type by quantifying adducts after exhaustive hydrolysis of an aliquot of plasma protein. I would like to explain now what these biomarkers tell us about the condition. In the discovery phase study, algorithm features carboxymethylysine and carboxymethylarginine were both increased significantly in ASD. CML is formed partly and CMA is formed totally from glyoxal. Glyoxal is a reactive metabolite formed in lipid peroxidation. So this was an indication that there may be disturbance of lipid peroxidation in ASD. Glyoxal is normally metabolized by the glyoxalase system. Prior evidence of increased lipid peroxidation in ASD was increased plasma malondialdehyde, MDA, and increased urinary isoprostanes. These are described in the given references. The level of dityrosine was increased about 2-fold in children with ASD compared to TD subject control. Dityrosine is mainly formed by protein modification by a dual oxidase, an enzyme that cross-link protein oxidatively. Increased duox is linked to neuronal plasticity in response to metabolic or environmental challenge. These biomarkers may be mechanistic, reporting on increased neuronal lipid peroxidation and neuronal plasticity. The only biomarker was found decrease in subjects with ASD was 3-DG hydroimidazolone, is a glycation adduct formed from the dicarbonyl metabolite 3-deoxyglucosone. 3-DG modified proteins have unexpected surface hydrophobicity and are unfolded. Their removal is mediated by activation of the unfolded protein response, UPR, a transcriptional system sensing the folded status of proteins, activating when unfolded protein levels are abnormally high to clear them by proteolysis with concurrent stimulation of low-grade inflammation. Decreased 3-DGH may be reporting abnormal activation of the UPR. Consistent with this, recent transcriptomic studies of post-mortem brain of ASD subjects found increased expression of chaperone genes for increased protein folding, reflecting increased activation of the UPR. So our blood test biomarkers report on disturbed metabolism may involved in the development of autism. Now the question you may have, why use plasma protein? Plasma protein is an accessible source of cumulative protein modification. Mostly 60% is serum albumin, which has a half-life of about 21 days. The protein accumulates modifications during this period and exchanges with albumin in the cerebrospinal fluid via lymph with a half-life of about 5 hours. We measure multiple chemically defined plasma protein glycation and oxidation adducts, which are established clinical measures of glycation and oxidative damage. And these biomarkers report on protein glycation and oxidation in the CSF and CNS neurons, related metabolic and functional disturbances close to the ASD phenotype. The advantage of using plasma protein biomarkers is that they provide a cumulative measure of lipid peroxidation over the lifetime of albumin, which is 3 to 4 weeks prior to sampling. So our analytical platform is stable isotopic dilution analysis, liquid chromatography, tandem mass spectrometry, LC-MS. This analytical technique is the gold standard reference technique for quantification of small molecules such as amino acids herein. It has high analytical sensitivity and specificity, robust calibration, and good reproducibility. Additionally, LC-MS is often preferred for harmonization of analytical measurements between laboratories and is regarded by the FDA as an appropriate analytical technology for Class II laboratory-based tests, such as likely applies to a blood test for autism. Pre-analytic processing involves exhaustive enzymatic hydrolysis. This is conveniently and reproducibly performed using a sample autoprocessor. We have described the method in detail in our recent paper in Assays in Biochemistry. We developed the method and have used it since 2003. Other research teams worldwide have applied it to their studies. We call this technique Ageomics. So in summary, a simple blood test for autism has been developed based on assay of plasma protein glycation and oxidation biomarkers, because these biomarkers accumulate and give you the clear changes in the condition. Discovery phase of 88% has been validated in a large independent international multi-center cohort with over 470 samples. Biomarker changes likely reflect activation of the UPR and neuronal plasticity in the brain in autism in response to risk factors during pregnancy. Why during pregnancy? For that, you need to see my next presentation.